Hi, everyone. Thank you all for being here. So today we're going to have a talk from Dr. Ayellet Segre, who is an assistant professor of ophthalmology at Mass Eye and Ear, Harvard Medical School. She's also an associate member of the Center for Genomic Medicine at the Massachusetts General Hospital. She's an associate member of the Broad Institute of Harvard and MIT and a coordinator of the Human Cell Atlas Bionetwork together with Rui Chen. Prior to joining Mass Eye and Ear, she was a lead computational biologist in the Genotype Tissue Expression Project, also known by some of you, I'm sure, as GTEx. There she played a key role in generating one of the most widely used genomic resources in the entire research community. Following undergraduate and graduate studies in life sciences and cancer research, Dr. Segre obtained her PhD in genetics and genomics from Harvard University. She then pursued a postdoctoral fellowship at Massachusetts General Hospital and the Broad Institute of Harvard and MIT, working primarily on the genetics of complex diseases. Dr. Segre's research reflects her highly multidisciplinary background, which includes molecular and cellular biology, computational and evolutionary biology, and statistical genetics and genomics. She applies her broad expertise to elucidate the genetic and biological basis of ocular diseases, including glaucoma, age-related macular degeneration, and diabetic retinopathy. Dr. Segre has played a crucial role in many of the landmark studies that have significantly furthered our understanding of these diseases. One of her strengths has been her ability to develop tools and platforms to integrate multiple data types. For instance, she recently published a paper in which she integrates genetic and expression data, including single cell, to narrow down IOP-dependent and independent regulatory mechanisms, genes, and cell types that may contribute to the pathogenesis of primary angle glaucoma. Plainly said, we're very lucky that Dr. Segre has decided to focus on ocular diseases. She's at the forefront of human genetics, single cell and functional genomics, and systems biology. She's the type of investigator who develops tools that make science easier for a lot of people, including for me. So it really is an honor to receive her here at the Moran, and please join me in welcoming her. Thank you.
Thank you for the kind introduction. I want to thank Dr. Zuach for inviting me and for the whole team for this great honor to present to you today. So no disclosures. So in my lab, we're interested in using human genetics to guide us to the causal mechanisms of common retinal diseases, retinal degeneration diseases in particular, primary open-angle glaucoma and age-related macular degeneration. And the reason for that is that germline genetics, by definition, implies causality and is not affected by the disease state for most cases, as opposed to other molecular modalities. And we're really lucky to be at this period of a genomic revolution where the prices have gone down for genome-wide genotyping with arrays, enabling to genotype hundreds of thousands to 1 million common DNA variants genome-wide in hundreds of thousands, and we're reaching even millions of individuals in the world from a range of diseases and health states. And the prices for whole genome sequencing have also significantly gone down to only several $100s an individual, allowing to query around 40 million both common and rare variants genome-wide. And this has really enabled over the past, I'd say, decade and a half, the acceleration of very large genetic studies, what we call genome-wide association studies, and I'll describe the study, the approach in a minute, which has been enabling the study of many diseases through different biobanks, such as the UK Biobank, all of us, whose goal is to perform whole genome sequencing on up to 1 million individuals. At Mass General Brigham, we have genotype data for over 60,000 individuals, and there's a consortium for global biobanks. So there's a lot of efforts around using genetics to study complex disease, and I'll show you soon why this could be very, very valuable, both for understanding the biological processes that lead to disease and for proposing new drug targets and therapeutic avenues. So what is a genome-wide association study? So I'm sure most of you know that most common genetic variation is a single nucleotide polymorphism. Change the laser pointer. Red instead of blue. But anyhow, the most common genetic variation is a single nucleotide polymorphism. We also have insertions and deletions that both the genotype arrays and whole genome sequence can query. And in a genome-wide association study, we're comparing these genetic variants genome-wide between a group of individuals that have a disease, with equal cases, versus individuals that do not have a disease. And in this toy example, this SNP, the GA SNP, there is a significant enrichment of the G allele in the cases versus the controls. And so this we would call an association with the disease. We use linear regression to identify these associations in complex traits, and logistic regression for dichotomous traits such as glaucoma. And in these models, we also correct importantly for covariates such as age and sex, or other covariates that may have an effect, such as differences in the population backgrounds, ancestral backgrounds. Okay, great, thank you. And so for each variant, we'll compute a test statistic and a p-value. And because we're testing on average 8 to 10 million common variants, and standardly a variant is considered common if its minor low frequency is above 1% of the population, we'll correct for about 1 million independent DNA variants, correcting for linkage disequilibrium, leading to a genome-wide significant cutoff of 510 to the -8. So linkage disequilibrium, it's an important term in the field of genetics, which just basically means that two variants that very close to each other in the chromosome, they'll tend to co-segregate from parent to offspring. And the further two variants are away from each other, the more likely that recombination will occur during meiosis, during the production of the germ cells. And the further variants are away from each other, the more likely we'll see different combinations of alleles. And the typical odds ratio of a variant found in GWAS, they're relatively small, between 1.05 to 1.4. A and D actually is an outlier where we have a few associations, an odds ratio between 5 and 10. And the majority of these odds ratios are 1.1, which, for example, if a disease prevalence is 4% in the population, one individual variant will increase the prevalence from 4 to 4.4. So these associations have been found to be largely additive. And so a genome-wide association study, one of the latest, largest cross-ancestry GWAS meta-analyses performed for primary open-angle glaucoma, was across around 34,000 cases and 350,000 controls, primarily made up of European, but also East Asian and African descendant individuals. And this GWAS has identified 127 loci in the region, throughout the genome, that reach genome-wide significance, together explaining about 9% of the heritability. So this suggests that there's still a lot of common variant associations of modest effects that have yet to be detected. And so just to orient you, this is a Manhattan plot. Each point here is a common DNA variant in the genome, so about 10 million points here. You're looking at the minus log p-value, the significance of each variant as a function of the chromosome position. And a very large GWAS meta-analysis has been performed led by Anthony Khawaja in the UK Biobank across about 140,000 individuals, primarily of European descent, has detected about 112 loci associated with IOP. There's high concordance in the effect size and direction between the variants associated with IOP and POAG, but there are also, as you'll see later, variants that are specific to POAG risk or to IOP. And these loci explain about 50% of the heritability of IOP. And very recently, from Stuart McGregor's lab in Australia, they've performed a multi-trait GWAS meta-analysis that meta-analyzed GWAS of POAG risk, IOP levels, and vertical cup-to-disc ratio. So traits that are either major risk factor or predictive of POAG across 600,000 individuals, and have more than double the number of loci that are associated with POAG risk to over 300 loci. So just to point out, glaucoma and these complex retinal diseases, glaucoma, AMD, what we're finding is that they're polygenic, just to kind of contrast that to Mendelian monogenic diseases, such as inherited retinal degeneration diseases. So these glaucoma is made up of multiple alleles of relatively modest effects compared to the monogenic Mendelian diseases, where it's typically one or most a few mutations, a very strong effect, and that lead to early onset versus more late onset disease. So there are many applications of GWAS, and starting with using the genetics to try and point us to the genes, and that may be leading to the disease susceptibility, that can point us to better understand the biological processes that lead to the disease. These can propose new potential drug targets, integrating with additional functional genomic data. As you'll see later, from the sequence alone, we can't easily always know what is the implicated gene or cell type. So integrating with single cell expression data, we can identify potentially the cell types in which these genes are exerting the causal effect on disease. These GWAS can also be used for diagnosis, for early prevention and intervention. You might have heard about using polygenic risk scores to calculate the genetic burden of individual developing a disease. So there's work that I've been doing with Dr. Jenny Riggs and Nazi Zebradas. I've been leading different projects. We can use it to distinguish disease subtypes for personalized medicine and to predict good responders to drugs through pharmacogenetic studies. So these are various utilities of GWAS that we've been working on. But today I want to focus on the first few, which is using the GWAS really to understand the underlying causal mechanisms, genes, pathways, and cell types that lead to disease. And why should we care, in particular, when I tell you that why should this potentially be relevant for therapeutics, when I tell you that each variant has a modest effect on the risk of developing the disease. So we and others have shown that these genetic associations, when tested in aggregate, they point to multiple genes that are enriched for targets of FDA-approved drugs for various complex diseases. We've shown this together with Jose Flores and David Altschuler for type 2 diabetes, and others for rheumatoid arthritis and other diseases. And there have been multiple examples now that are emerging from GWAS, where they're pointing to genes that either in retrospect are targets of known drugs for the disease, or a classical example is the HMG-CR, the HMG-CoA reductase, is the rate-limiting step in LDL cholesterol biosynthesis, and it falls in a GWAS locus both for LDL cholesterol levels and for myocardial infarction risk. It has a relatively small odds ratio of 1.1, but it is the target of the commonly used statins. Or the IL-12B cytokine that falls in the psoriasis GWAS locus, and there's a target antibody against the cytokine that suppresses the immune response. And there are many other examples, many of which are summarized in this review. And finally, also has been shown that target genes that are implicated by human genetic studies significantly increase the likelihood of success of clinical trials. So just a few words about primary open-angle glaucoma and the problems and questions that we're interested in addressing. I think this is very broad, but just kind of to give some context to the molecular and cellular questions we're asking. So as you all know, POAG is one of the leading causes of irreversible blindness, affecting over 70 million people worldwide. And it's characterized by a gradual retinal ganglial cell death and a progressive degeneration of the optic nerve that eventually leads to primarily peripheral visual field loss. And a major risk factor is elevated intraocular pressure, the blockage of the drainage of the aqueous humor, fluid through the outflow pathway, particularly through the trabecular meshwork, is thought to be one of the major contributors to elevated intraocular pressure. And there are no cures to date, and the best treatment are IOP-lowering medications. And to my understanding, they work pretty well, but they're not effective in all patients. And furthermore, kind of to add to the complexity of POAG, about 40% of POAG patients, their intraocular pressure is in the normal range, what we call normal tension glaucoma, suggesting that there are also IOP-independent mechanisms that may contribute to the development of POAG. And so there's really a strong need for neuroprotective drugs. And the good thing for us is that POAG is, I think, one of the most heritable diseases. Its heritability is estimated to be around 0.7 to 0.8. So we can, use the genetics to potentially uncover new causal mechanisms. And so some of the open questions we're interested in addressing are for the IOP homeostasis, what are the genes and cell types in the conventional and unconventional outflow pathway that are affecting IOP levels, homeostasis, and that lead to elevated IOP? And in the posterior segment, what are the genes and cell types that lead to that affect RGC sensitivity? Is it primarily dysfunction of genes in the RGCs themselves, or in neuronal support cells, such as non-neuronal cells? So the big question is, how do we go from these DNA variants to the implicated causal genes, pathways, and cell types? And there are several challenges to doing so. I messed up the pointer again. So the first one is, as I mentioned, because of linkage disequilibrium, these genetic associations point to often large genomic regions that contain multiple genes. On average, 10 to 20 genes are found in these large GWAS loci. So what you're seeing here, it's called a locus zoom plot. We're looking at the minus log p-value. Each point is a variant as a function of the chromosome position. These lines below are different genes, and the colors are color-coding these points, these variants, based on their linkage disequilibrium to the lead GWAS variants. You can see there's a lot of variants on top that are red, that are in high LD. So it's hard to distinguish, which is the actual causal variant in this region. Furthermore, the majority of these GWAS variants are found to be in non-coding regions. So you think of the central dogma from a gene going, a DNA encoding an RNA, encoding a protein. These variants seem to be affecting the first step, the expression, the regulation of the gene from DNA to RNA, more so than affecting the protein structure function, as the majority do not fall in exons, but either in introns or in intergenic regions. And so this suggests that the majority of these GWAS variants are going to be affecting disease through regulatory mechanisms. And some of these are shown here. They could be affecting the promoter of a proximal gene. They could be affecting an enhancer that regulates a gene that's more distal. So it's not necessarily the nearest gene that's the affected gene, or they could be affecting the alternative splicing, yielding different isoforms and different protein products. Furthermore, the genetic regulation effect can vary by tissue and cell type, and we've seen this in different studies, as I'll show you later. So we really want to measure these genetic regulatory effects in the relevant tissues and cell types. And so how do we detect these genetic regulatory effects? So demonstrating this on the eye, we isolate the relevant tissue, extract the RNA from the tissue. This is just a very high-level schematics, perform RNA sequencing, perform multiple kind of extensive quality control on the samples, and then quantify gene expression levels and alternative splicing. At the same time, we extract either DNA from the blood or from the retina from the tissue. We perform either genotype array and imputation. Imputation is basically statistically inferring the common variant pools for the, since arrays only genotype directly 500,000 to 1 million variants, we can infer the rest of the 9 to 10 million variants from a reference panel of whole genome sequence haplotypes from a matched population. There have been a lot of methods that have been developed and perform really well. Or we perform whole genome sequencing to get the full query of variants genome-wide, and then again QC and variant calling per donor. And as you'll see in the next slide, we need to perform RNA sequencing and DNA sequencing on at least 70 donors per sample to have enough statistical power to detect these genetic regulatory effects. And that is because the most common approach to detecting these genetic regulatory effects are called expression or splicing quantitative trait loci. You're basically correlating the genotype at a given variant position with the expression or splicing levels of a gene. Looking at all the variants within a cis window is kind of arbitrarily defined as 1 megabase around the transcription start site of a gene. And for each variant, we will check if there's any correlation of the genotype across a population of individuals. So for statistical power, we need at least 70 to 100 different individuals to get genetic variation. And variants that show, like in this toy example, where the T leads to high expression levels and the A low expression levels, or the T leads to the long isoform versus the short isoform. These are what we call expression of splicing QTLs. And this is a linear regression model where we correct also for covariates, including ancestral background using genotype principal components. So these expression QTLs have been found for over 10,000 genes in the human peripheral retina. This is from a study from Anand Swaroop's lab from NEI, where they performed RNA sequencing on 400 peripheral samples from 400 different donors. And we have been collaborating with them to also compute expression and splicing QTLs in the macula from 185 samples in the macula as well as periphery. And these results will be made available soon. But the work I'll show you today is using the peripheral retina AQTLs. But in addition, expression and splicing QTLs have been computed for almost 50 tissues in the human body. This was part of the Genotype Tissue Expression Project, where whole genome sequence was performed on over 800 donors, and RNA sequencing was performed on 70 to 700 samples from 49 different tissue sites in the human body, including 11 sections in the brain, artery, nerve, blood, muscle. And this had led to the detection of EQTLs in over 15,000 genes and splice QTLs in over 5,000 genes. And you can see that the discovery power scales with sample size. So the more samples you have per tissue, the more you're able to detect genetic regulatory effects of weaker effects. And EQTLs have been discovered for most of the protein coding genes and for 80% of the long non-coding RNAs. So why should we care about the regulatory effects found in non-ocular tissues for the purpose of studying eye diseases, common retinal diseases? That's because if we look at the distribution of the tissue specificity of these EQTLs and SQTLs, we can see that it's bimodal. So about 70 to 80% of these QTLs are shared across multiple tissues. And that's partly due because there's sharing of cell types across tissues, and also because some of the genes and regulatory effects are functioning in multiple tissues. And many of the GTEx tissues contain cell types that are relevant to glaucoma, such as artery, fibroblasts, and brain that contains both neuronal and micro and macroglial cells. So this suggests that we can use the ENSQTLs found in the GTEx tissues to study the regulatory effects that may be explaining some of the associations with glaucoma. And that's also a question we posed when we started this work, but also suggests that 20 to 30% might be tissue specific, and so it's important at the end of the day to measure these regulatory effects in the relevant tissues, in addition to retina, the outflow pathways, the optic nerve head, and we have a study ongoing towards that goal that I'll mention at the end. So the first part of the work I'll show you today is testing whether we can use these EQTLs and SQTLs from the retina and the 99 GTEx tissues to try and interpret and propose what may be the underlying causal regulatory mechanisms and genes implicated by the 127 POAG variants and 133 IOP variants in the 112 loci. Just to add more complexity, if you look at it in a GWAS locus, most GWAS loci have actually multiple EQTLs or SQTLs that fall in the region that are acting on multiple genes. So for example, on average, looking at the POAG or IOP, GWAS is on average 17 E genes are being regulated by variants in the LD interval around the GWAS associations, or 7 S genes, 7 genes that are being regulated by SQTLs. So how do we know if any or all of these E or SQTLs are the underlying causal mechanism? So we applied a Bayesian approach called the colocalization analysis that basically, just very high level, tests whether two co-occurring association signals, a GWAS signal and a QTL signal, if they're tagging the same causal variant or not. This Bayesian approach takes into consideration the GWAS statistics, the local LD, allelic heterogeneity, and there's some fine mapping, and it basically produces a posterior probability that they're tagging the same causal variant or not. And this is an example. This is the minus log p-value of the GWAS versus that of the QTL. This is an example where they're each tagging a different causal variant, an example with this correlation, and they're tagging the same causal variant. And a positive result suggests that the expression of splicing QTL may be the underlying causal mechanism of the genetic association. So when we applied it to the POAG and IOP loci, we found significant colocalization for about 60% of the loci. So about 200 genes mapped to 76 loci for POAG. And these were enriched in the following biological processes, including elastic fiber formation, extracellular matrix organization, the TGF-beta signaling processes, which have been implicated previously, both from previous GWAS studies and functional studies, experimental studies, and enriched in retina-related processes, looking at the mouse phenotype ontology processes. We saw enrichment of genes that affect the retina morphology. The IOP genes, loci, 279 genes were suggested for 79 GWAS loci, and these were enriched in, the strongest enrichment was target genes of this VENTX transcription factor that's involved in development and hematopoiesis. And I'll say a few more words about what the target genes may be doing, as well as in vasculature development, regulation of cytoskeleton organization, and cell adhesion. So this work suggests both IOP-dependent and independent associations with glaucoma, and these are some of the genes that fall in some of these loci. So this is suggesting, and as you'll see, this kind of ties in later when we start looking at which cell types these genes are clustering and expressed in. This may suggest that the extracellular matrix organization in the outflow pathway might be affecting the drainage of the aqueous humor. But as we'll see, there's also involvement of these genes in the posterior part of the eye, in the optic nerve head. So if we look at the target genes of the VENTX, we find that they're most highly expressed in immune cells and vascular cell types. So these are the target genes, and this is looking at their expression levels. Red is high expression, and the size of the dots represents the percent of cells that are expressed. And this is single nuke RNA-seq data from Josh Sain's lab, and these tissues were actually collected from Greg Hageman's and the Moran Center's sample repository. And so you can see that they're largely enriched in lymphocytes or macrophages, as well as in vascular cells, parasites that may be affecting the blood flow, and the Schlemm's canal that may be affecting the aqueous humor fluid. So this is some kind of new suggestions that some new genes that are suggested to be potentially involved in an immune-related process for regulating IOP. And to show you a few examples of what we can learn from these expression and splicing QTLs, this is an example of an IOP-dependent mechanism. So EFMP1, it's an EGF containing 5 billion extracellular matrix protein. You can see that this gene actually doesn't fall underneath the lead GWAS variant, but it's actually 100 kb downstream. So just by looking at the genetic region labeled by kind of marked by the GWAS association, we would have not necessarily prioritized this gene. But there is a variant here that regulates the splicing of this gene that's correlated with the genetic association and has a significant colocalization posterior probability of 0.5. And this work suggests that the skipping of exon 6 and 7 may be protective for POEG. So it produces a protein that decreases risk of POEG. And so we're interested in further understanding what are the protein domains that these exons are affecting. And I've been having discussions with Dr. Janie Wiggs of testing this maybe in a model organism such as zebrafish. And interestingly, in a study that Janie Wiggs and Ryan Colantis have performed in a Filipino family, where they performed whole exome sequencing on a familial juvenile onset open glaucoma family, they found an enrichment of rare deleterious mutations in this gene that seems to be associated with the juvenile onset of glaucoma. So here, this is an example where we're finding an intersection between kind of more deleterious rare variants falling in a gene that can lead to early onset glaucoma. And this is a gene involved in extracellular matrix, but variants that maybe have a more milder effect on the protein function, such as alternative splicing, that's found in the natural population, may lead to a later onset of the disease. The EQTLs can also provide information on the direction of effect, which can be important for both understanding the biological mechanism, but also for drug development. So this is an example where an EQTL that affects angiopoietin-1, and this was found in the brain, cerebellum, increase of expression of this gene is suggested to decrease intraocular pressure, and angiopoietin-1 has been shown to play an important role in vasculature development. And it's required for the studies shown in mouse and human that is required for Schlemm's canal development. And just to show you one example of an IOP independent mechanism, this is an EQTL found in the nerve in GTEx that co-localized with the POEG GWAS signal. And it suggests an increase of RE. It's an arginine glutamic acid dipeptide repeats gene, increases risk of glaucoma. And this gene triggers apoptosis. It's a transcriptional co-repressor during embryonic development. And it's found to be expressed in retina and the optic nerve head in human tissue. And it lies in a locus that is not associated with intraocular pressure. And interestingly, when we inspected this 3D conformation of chromatin map that was generated in 5 different human retina samples in a Nancy Roop's lab, we found that, so this is a technique I see that can detect physical contact in the 3D space between very distant regions in the chromatin along the chromosome. We found that the POEG variant that falls 400 kb away from the RE transcription start site, it forms a physical contact with the start site of the RE, providing additional support that this indeed may be the underlying causal mechanism for this genetic association. So the next question we're interested in asking, thinking both about better understanding the underlying biology and for therapeutic design, is in what cell types and what tissues in the eye are these genes that have been suggested to be implicated by the GWAS loci based on the EQTL and SQTL mapping, in what cell types maybe they be exerting their causal effect. And the underlying assumption is that disease-associated genes are going to cluster in particular cell types in relevant tissue, and that would suggest that that cell type may be pathogenic to the disease. in an ideal world, we'd actually want to measure these genetic regulatory effects in particular cell types in the relevant eye tissues, and we're doing work towards that goal, but we're still, this is still a long way to go. And so in the meantime, since, let's see if I have the next slide, but, whole human eye cell atlas, and this is just one example from Josh Sain's lab, but there have been many other labs that have been performing single cell and single nuc RNA sequencing on many tissues in the eye, both from the anterior segment, the cornea, lens, iris, ciliary body, trabecular meshwork, corneal scleral wedge, and then in the posterior, both peripheral and macular retina and RPE, and the optic nerve head and surrounding tissues. But these have been performed in a handful of donors, maybe for the retina and a larger number. And as you saw, just go back, to compute EQTLs, we really need a large sample size of 70 to single nuc RNA sequencing from 70 to 100 different donors. So in the meantime, we started by using just gene expression in single cell data, to test whether genes that map to GWAS loci enrich for expression in particular cell types. And I'm not going to go through the method, we call it Eclipser, but it basically uses a Bayesian-Fisher's exact test to test whether the distribution of the cell type specific scores of GWAS loci based on their genes that map to them, if they're enriched compared to GWAS loci of unrelated traits. And you can find more details on both in a publication and a preprint online. And so just as a proof of principle, we tested this method on 21 traits in 8 GTEx tissues for which single nuc RNA-seq was performed, and found enrichment in relevant cell types for diseases where this cell type is known. So for example, genes that map to GWAS loci for atrial fibrillation were enriched in myonuclei in heart, or genes associated with prostate cancer were enriched in a cell type of origin, luminal epithelia in prostate, and melanoma and melanocytes in the skin. So that gave us confidence that this approach, testing whether genes that fall in GWAS, that are mapped to GWAS loci for a given complex disease, if they're enriched for cell type specific expression in a given cell type, can point to relevant pathogenic cell types. So we applied this to the 6 tissues from the anterior segment, including the trabecular meshwork and Schlemm's canal and the ciliary body and iris, where there's 39 cell types have been detected based on clustering of the gene expression in these cell types. We applied it to the both peripheral and macular retina, where 16 cell types have been detected, and to the optic nerve head, optic nerve, peripapillary sclera, sclera and choroid, where 36 cell types have been detected. And these single nuc RNA-seq study, both in the posterior part and the anterior part that I'll show you today, is from Josh Sain's work in collaboration with Greg Hageman. As I mentioned, a lot of the tissues were from the Lions Eye Bank here at Utah. And the retina is from Ray Chen's lab, from the periphery, and the macula from Josh Sain's lab. So just show you some of the results. I'll orient you on one of these bubble maps. These are the size correlates with the significance, the minus log p-value, and the color correlates with the fold enrichment within a given cell type. Each column here is a different cell type. And these are the GWAS loci for POAG from the cross-ancestry GWAS. These are for the IOP. And then we also looked at just the European subset for the POAG GWAS. And what we found was significant enrichment of genes that map to POAG in the fibroblasts that fall in the ciliary body, in the iris, and in the trabecular meshwork. Genes that are associated with intraocular pressure were also enriched in parasites and lymphatic endothelial cells, vascular endothelial cells, and nominally in the Schlemm's canal. So this suggested to us that the enrichment that we found was actually a bit stronger in the ciliary body and iris compared to trabecular meshwork. And so this suggests that the uveoscleral, the fibroblasts in the uveoscleral may be, contributing to the IOP homeostasis, as importantly as the trabeculum fibroblasts and the trabecular meshwork. Looking at the retina, we found that the genes that were mapped to both POAG and IOP, but primarily to POAG, were enriched in macroglial cells, in astrocytes and euloglial cells. And we did not see enrichment in retina ganglial cells. Now, this doesn't mean that genes that are changes or effects on the function or regulation of genes in the retina ganglial cells are not affecting its sensitivity. It just means that based on the functional genomic data we have to date and our mapping of genes to GWAS loci for POAG, it seems that the primary dysregulation that's affecting POAG is coming from these astrocytes and euloglia cells. And it could be also that these GWAS are highly enriched for individuals of high tension glaucoma. And we actually have a ongoing study that Dr. Genomics is leading to perform a GWAS in normal tension glaucoma to see if we can find, GWAS loci for NTG that may be potentially more enriched in retina ganglial cells. And so there have been several studies in animal models that show that astrocytes and euloglia cells become reactive at early stages of the glaucomatous eye in mice when the RGCs are still intact. And the astrocytes are important both for the structure, metabolism, and function, the neurotransmission, and the vasculature development of the retina. So this proposes a lot of potential functions that may be causal to the RGC sensitivity, from nourishing it to supporting its structure, or even affecting the blood flow to the optic nerve. And just to summarize the results in the optic nerve head, we found significant enrichment also of POAG genes in the astrocytes and oligodendrocytes in the optic nerve head and the optic nerve. And then genes associated both with POAG and IOP were enriched in fibroblasts, primarily in the peripapillary sclera, that we can think that might be affecting the structure or the mechanical stress on the optic nerve, but also in the choroid and the optic nerve head, and also enrichment in vascular endothelial cells in the choroid and optic nerve head that may be affecting the blood flow to the optic nerve. We can also then delve deep into these enriched cell types and ask what are the genes that are driving the enrichment in these cell types to gain some insight on potential cell type specific gene targets. And so what you see here is the fold change, the cell type specificity of the genes in astrocytes compared to other cells in the optic nerve head, the DGKG, disulfoglycerol kinase gamma, that functions in sterol and lipid binding, is the most specifically expressed in the astrocytes, followed by many other genes. And this DGKG was found based on a retina-specific EQTL. So this is one example where having the EQTL in the relevant tissue was important. And we find that these genes are enriched also in extracellular matrix organization and TGF-beta signaling. So posing some lipid-related and extracellular matrix-related processes in astrocytes that may be affecting the RGCs. So just to conclude, I hope I showed you that using large human genetic studies together with functional genomic data can propose genes and biological processes that may be important for the development of glaucoma, both IOP dependent and independent. We found a potentially new link between immune-related processes and IOP through the Ventex transcription factor. Our work suggests that the unconventional outflow pathway may be kind of underappreciated with respect to its role in regulating IOP. And to my understanding, I'm not a clinician, but my understanding is that most of the IOP-lowering medications are targeting the trabecular meshwork. But we might want to think also of developing further therapeutics that target the ciliary muscle. Parasites have been implicated in IOP regulation that maybe affect the blood flow in the anterior segment. And we found that neuronal support cells, both in the retina and the optic nerve head, such as astrocytes, fibroblasts, and oligodendrocytes, may play a prominent role in retina ganglion cell sensitivity, potentially affecting their structure, function, and vasculature. And so I think this is important to consider in thinking about targeted gene and cell therapy, especially as people are developing regeneration of RGCs or cell therapy, thinking about also targeting at the same time the macroglial cells, for example. And so this work also created a lot of hypotheses that need to be functionally experimentally followed up. And so the next steps, as I had mentioned, the ENSQTLs from retina and from the 49 non-ocular tissues have suggested multiple genes for about 60% of the GWAS loci, but we still don't have the full picture. And also I did mention that on average, the ENSQTLs suggest about 3 to 4 genes per locus. And so there's still a lot of open questions as to whether this is because we're not yet looking at the correct tissue or cell type, or because we know that there is also a lot of co-regulation of genes that are adjacent to each other. And it could be that each genetic association is actually affecting the expression of multiple genes that have an effect on the disease. And so our next step is to try and detect genetic regulation, both in the anterior and posterior healthy eye tissues that are relevant for glaucoma at the tissue level and the single cell level. So we have collected 10 different eye tissues from 100 postmortem donor eyes and have performed whole genome sequencing on their DNA and bulk RNA sequencing on the optic nerve head macula, which is more enriched for RGCs than the peripheral, the ciliary body, and the trabeculometric Schlemm's canal. And in collaboration with Tim Cherry, we've also working on performing a TAC-seq that's to identify open chromatin regions that can suggest regions that have transcriptional activity. And so we're hoping this year to have also expression splicing QTLs for more relevant tissues for glaucoma and see if we can shed additional light on the causal mechanisms. Maybe then we'll also find some of the genes are enriched in RGCs, and we might find some additional mechanisms that haven't been exposed yet. And then towards getting at the single cell level, in collaboration with Josh Sains, we're working on initially looking at allele-specific expression in the single nuc RNA-seq data that we have from the 10 different tissue regions from 17 donors for which we have performed whole genome sequencing. And eventually, together with Ray Chen, has also been doing a lot of efforts in performing single nuc RNA-seq on over 60 donors for each of these tissues. I think eventually we will have single cell RNA-seq from enough donors to be able to look at expression and splicing QTLs at the single cell level in relevant eye tissues for glaucoma. So I think we're still at a exciting point where there's still, I think we've made some potentially informative discoveries, but there's still a lot to be learned. And just I think my last slide and some future considerations to kind of enhance these type of studies with genetics and functional genomics is to really think about the detailed ocular phenotyping and collecting blood samples when you can for genotyping or DNA sequencing from patients. I think some of the, especially PRAG is a very heterogeneous disease, and if we'll be able to do genetics on disease subtypes, as I mentioned, Dr. Jane Riggs is leading a normal tension glaucoma study. This might help identify biological processes or targets that may be relevant for one group of patients versus the other. Other important efforts is doing these genetic studies on larger samples for diverse ancestral backgrounds. And for example, Dr. Jessica Cook-Bell is doing great work on performing PRAG in African ancestry, and Joan O'Brien has also published, and Mike Hauser studies in African GWAS. And we started looking at in collaboration with Jessica Cook-Bailey, we started looking at the genes, in which cell types of genes that map to the African GWAS loci. I can't give a spoiler, but we are finding some potential differences. And so I think really having genetic studies of diverse ancestral populations will be very important for kind of clinical care to all of our patients. And then finally, performing functional genomics of the relevant eye tissues at not only the single cell, but also spatiotemporal resolution, looking at other modalities such as protein expression and epigenetics, and the samples that we performed both for GTEx and retina and the single cell experiments were primarily from European descendant, and so I think it's also important for us to perform these in other ancestral backgrounds. Ray Chen has been doing this for the single nuke RNA sequence. I think we're going to have functional genomic data from different ancestral background samples, which might also help shed light on similarities and differences in causal mechanisms. So with that, I just want to thank my lab, and in particular the members that have performed the bulk of the work I presented to you today, and then all the wonderful collaborators without whom we couldn't have, our lab and the collaborations could have been possible to do this work, and the funding agencies. So with that, I'm happy to stop and take questions.